joining us for today's Keeping It Clean in 2015 webcast, Germs in the News, What Caused Illness in 2015. Last few participants are logging in. I would like to highlight some housekeeping items for today's session. The brief is being recorded and all participants will be in listen-only mode. All privacy concerns, the attendee list will not be displayed. If you're within WebEx, please refer to the chat box on the right side of the screen where I will add some technical help information, including a WebEx technical support number. If you are panelists, please type into the Q&A box located on the right side of the screen. Please ask option at all panelists. We do invite you to submit questions at any time so they may be queued. We will be short video. The video will play through your computer speakers, so please adjust them accordingly. Ladies and gentlemen, I would now like to turn the presentation over to your moderator today, lead scientist with Ecolab, Dr. Anna Starobin.
Good afternoon, everybody. Thank you for joining us. The topic of today's webinar is Keeping It Clean in 2015, Germs in the News, What Caused Illness in 2015. Our speaker today, Dr. Ruth Petran, Vice President of Food Safety and Public Health at Ecolab, will discuss how microbes are getting coverage in the news, including Listeria, Salmonella, and Cephalospora, and some other topics. Dr. Petran will also explore the epidemiology, ecology, risk management, strategies and outbreak examples of illness, and she will cover today these topics. As Vice President of Food Safety and Public Health at Ecolab, Dr. Petran provides technical expertise and consultation to internal and external customers on food safety issues, identifying and tracking emerging trends and new control strategies. Please welcome Ruth to her presentation. Thank you.
Thank you, and I am pleased to be speaking to all of you. However, I have just had a technical glitch, and Tyler, I'm going to have to ask you to change the slides for me as I tell you. I have lost my WebEx connection, but fortunately my phone is still working, so I think we're okay then. I hope everyone can hear me.
Petron, go ahead.
Thank you. So today we will be talking about those sort of newsworthy germs that we have heard about in the last several months. We've had this as a requested topic from feedback from attendees to these webinars. So I just want to touch on some of these through this webinar. So next slide, please. Essentially, we will focus on 4 different newsworthy events that occurred in 2015. We'll talk about listeria in dairy products, salmonella in cucumbers, cyclospora in cilantro, and then a general category of illnesses attributed to temperature abuse. Slide, please. But to stage, I want to start off with an overview of some food safety facts and figures. Picture of foodborne illness on an annual basis around the world is a little disheartening. It's estimated each year that 582 million people get sick, and perhaps 350,000 or more of them die each year due to foodborne illness. In less than most of the developed world, this translates into about 1 in 6 or 8 of us become sick each year because of foodborne illness. Clearly, these numbers are too high. Next slide, please. What kind of agents make people sick? Well, data from the U.S. CDC come out each year and represent the most recent data that we have through 2014. And you can see that these are reported by numbers of cases per 100,000 people, which is typically how illness rates are reported. Salmonella continues to be the top bacterial foodborne illness, clocking at about 15 cases per 100,000 people. There were notable changes in these data this year. First, in E. coli 0157H7, whose numbers dropped this year, and those Shiga toxin E. coli's that are not 0157 strains increased this year. If we move to the right-hand column, the 2020 National Health Objectives, we see that generally most of these illness rates are higher than we should be. So again, there's big opportunities for improvement in food safety. Next slide, please. We know that the age of the person who gets sick makes a difference. To summarize some information showing that the incidence of illness is most likely to occur within young children, that is less than 5, but the more serious kinds of complications, including hospitalizations and deaths, are more likely to occur in those people 65 years or older. And the inevitable aging of the population impacts this significantly. Please. These illustrate the types of that recalls occur in the U.S. And we see that for the green piece of the pie, undeclared allergens are number 1, both with FDA-regulated products and USDA-regulated products. We see incidents recalls attributed to the pathogens of Salmonella and Listeria monocytogenes appearing next on the list. So clearly, issues related to microbial hazards continue to be a concern. Slide, please. Where to focus efforts to address these microbial hazards? Data, also from CDC, illustrate that the majority of reported illnesses in the U.S. are attributed to food service type environments. So again, opportunities to focus food safety efforts certainly need to be directed here. We can't ignore the rest of the supply chain, but certainly efforts need to be directed to food service as well. Next slide, please. How do we direct these efforts? A set of data from CDC, shown here, illustrates those factors that contributed to foodborne illness in 2013. And we see the whole category of improper holding, which relates to temperature control, the number one contributing factor to illness. We also see issues attributed to poor personal hygiene, causing about 29% of outbreaks, and issues related to unsafe source and contamination from the equipment and environment next on the list. This gives us a clue as to where to direct risk management strategies for food safety. Slide, please. And lastly, to drive this point home a little bit further, it was published a couple of years ago, specifically looking at contributing factors to these restaurant outbreaks. And here we see the impact of poor personal hygiene habits, behaviors really contributing significantly with handling by an infected person and ban contact the majority of the issues. We also see concerns related to temperature and cleaning that need attention as well. Slide, please. Jump into the meat of this presentation with that as a background. First, focusing on Listeria, which is a formidable enemy that we have seen for many, many years as concern with in our food safety realm. Well, like most bacteria is something that can't be seen or smelled or tasted. We know that it is able to survive a wide range of temperatures, generally from refrigeration temperatures up to fairly warm temperatures, although its optimum temperature tends to be about the 86 to 98 degree Fahrenheit range. Able to survive over a wide pH range. And salt tolerant. We see that it can survive as high as 25% salt concentrations. It has a low water activity tolerance, so those foods with less moisture in them still allow survival of Listeria. It is able to survive under dry conditions for long times and in the freezer as well. Next slide, please. Where about Listeria? Well, firstly, it causes a lot of illness. It is a very costly foodborne pathogen in the U.S. We'll talk about salmonella in a little bit, but clearly the fact that it causes $2 billion in mean annual costs of health care is significant. It has a very high hospitalization rate. Mortality rate is very high. About 20% or more of cases affected with Listeria will die. And the people who are most at risk for Listeria infection include pregnant women, fetus, newborns, the elderly, and anybody who's immunocompromised. High mortality rate leads to a high cost per each case of illness at more than $1 million. Next, please. Now, the challenge with Listeria is that it's very commonly found in soil, water, plants. Humans can carry it. Some animals. We know it favors cool and moist conditions. Frankly, most of us are probably exposed to Listeria with very little health consequences. Very virulent strain of Listeria is Listeria monocytogenes, and this is the strain that causes the most serious illnesses. It's a positive rod-shaped bacteria, and the picture shows what it looks like under a microscope. It grows at cold temperatures, so it's psychotrophic. It can grow with or without oxygen. It doesn't form spores, and it can produce biofilms. Next, please. This shows the overall incidence of Listeria monocytogenes cases over the last couple of decades. And it shows that from a high back in the late 80s, about 8 cases per 100,000, we've significantly, if you just look at the last, say, 10 to 15 years, generally the cases have leveled off. And this is concerning, certainly from a public health standpoint, but also to regulators. So there has been a lot of risk assessment done in order to study this further. Next, please. The 2000s, a joint risk assessment project was pulled off and done by the FDA and FSIS, or the Food Safety Inspection Service, which is part of USDA, to come up with these relative risk rankings of these slides. Certain foods were categorized into high, moderate, and low risk. You could click one forward. There will be a bill on this slide, please. To note that recent recalls and outbreaks have shown that Listeria can survive and produce illnesses, really any of these products, regardless of where they may reside on this overall risk ranking that was done again 15 years ago. This is something that the regulators are potentially re-looking at, so we need to keep our eyes on where this may go. Next slide, please. So from these risk assessments, we identified several contributing factors which were associated with a higher risk of listeriosis from ready-to-eat foods, and related to the amount and frequency that a food was consumed, often and how highly it was contaminated, whether that food could support growth, the nature of that food, and how long it was stored for. Risk were, frankly, a small proportion of foods that were contaminated with relatively high levels of listeria. Please. Since the late 80s, in the U.S., we have had these regulatory policies established against listeria, essentially a zero tolerance by both regulatory agencies, which means that if any Listeria monocytogenes is detected in kinds of foods, the food is considered adulterated. What are some of the outbreaks that Listeria has caused? And I'll look back in history a little bit and then give a present-day approach as well. I'm eyed with Listeria monocytogenes historical outbreaks. The first outbreaks we heard about related to Listeria, a Mexican-style cheese back in the mid-'80s. Up until recently, was among the deadliest of all foodborne illness outbreaks in the U.S. In this particular outbreak, there were about 140 cases identified in California, and about a 30% mortality rate, generally causing stillbirths and issues in premature babies. Associated with this was consumption of cheese from a particular manufacturer who, when it was investigated, was found to have a product environment, as well as product that tested positive for the outbreak strain. Big outbreak occurred about 15 years after that, attributed to hot dogs. In this case, about 100 people got sick, and there were at least 15 deaths. It was found in unopened packages of the hot dogs from this particular plant, as well as in the plant environment. And hypothesized that a construction project in the plant probably caused release of the organism into the processing areas. This is history. So let's look forward, and I would go to the next slide, please, of our recent outbreaks, because there were a couple earlier this year that occurred in ice cream. In this case, the number of cases was relatively small, about 10, but there were 3 deaths. So again, a very high mortality rate. With this one is the cases ranged over a time period of about 5 years and were associated with a certain brand of ice cream manufactured at several facilities. Through its investigation, there were several concerns identified with sanitation concerns, maintenance issues, sanitation, and personal hygiene that likely contributed to the contamination. Another outbreak that occurred in 2015, attributed to Listeria monocytogenes, was in soft cheeses. In this case, 30 people got sick, 28 of them were hospitalized, and 3 of them died. In this case, the illnesses were associated with soft cheese manufactured at a facility in California. And isolates from the plant environment closely matched those from the pens over a range of about 5 years. And the here from CDC shows this range, with cases ranging back to about April or May of 2010 through about July of 2015. Next, please. So let's hear for a minute on a new technology that is being used very widely in foodborne illness disease investigation, and this is the technology of whole genome sequencing. And this is something that has come to play and to use much more recently, and reflects and was used in these most recent outbreaks. Sequencing is a laboratory technique that reveals the complete DNA of an organism, that allows us to better understand variations both within and between species. It allows us to differentiate between organisms with very high precision that has been allowed to this point. This technology to identify pathogens which might be isolated from food, environment, and then compare these to samples from patients. And a match is made with a very reliable link between the two, which can help define the scope of these foodborne illness outbreaks. And here's where we see the effect of whole genome sequencing technology and its power in being able to identify outbreaks that occurred over a number of years in most recent Listeria outbreaks. So this is definitely a technology to keep our eyes on, because I think with its widespread use, we have the potential to see more of these. As we go to the next slide, please, how to manage Listeria. Next, please. And really think about Listeria control, we have to think about risk reduction. And this is a build slide, so if we could do the first click, please. Strategy is really to pay attention to what the risk assessments say, that a small percent of foods are likely contaminated. Next build, please. So the key to risk management is consistently applying Listeria control measures. Click, please. And includes modifying the food where possible. Check pH maybe, or the moisture content of the food, adding an antimicrobial substance. Next, click, please. Modifying extrinsic factors, that is in the environment of the food, changing the temperature at which something is stored. Next, click, please. Implementing controls through heating, root cleaning, and sanitizing. Next, please. And finally, preventing the establishment of niches within which Listeria monocytogenes could be harbored, because niches can serve as reservoirs that can essentially disperse the food, the organism rather, to food items. The effective sanitation, all of these niches, is key. Next slide, please. Here's to Salmonella. Salmonella also produces a very severe, rarely fatal illness. Symptoms include typical gastrointestinal type symptoms, and it can last for up to 5 days. The onset time is between 18 and 36 hours, but it can be shorter or longer. We have seen incidents where low numbers of Salmonella cells have caused illness. Showed on the previous slide, the incidence in 2014 was about 15 cases per 100,000 people. Next, please. Salmonella is the most costly foodborne pathogen in the U.S., with an estimated cost of $3.7 billion, millions of cases annually, and a pretty significant cost per case, although it's not as high as Listeria, given the lower fatality rate. Next, please. Salmonella in food. It for a relatively wide temperature range and moisture content as well, or water activity level, although not quite as low as Listeria. Freezing is not effective at eliminating Salmonella from a food, and survive long periods of dehydration. So that Salmonella is pretty heat resistant. We see a wide range among strains, and there's up to 2,000 different kinds of Salmonella. does depend on the food, its pH, its water content, etc. Next slide, please. And this list shows the so-called salmonella-sensitive items, or those kinds of foods that have been associated with illnesses or be linked to salmonella by virtue of the kind of food that it is. It does come from animals, so any meat or egg product or dairy product, for example, could be contaminated. But we also have issues where contamination from animals might get into other kinds of foods, in seafood, in produce, in the production of agricultural items like grains, chocolate, and peanut. Slide, please. With listeria, it's helpful to look back at some history, look to present day as well. So first, focusing on some history related to salmonella. We have seen outbreaks in many kinds of produce. Tomatoes have been contaminated a few times, and one thing we learned from one of these outbreaks is that salmonella can actually be internalized inside the tomato, providing the acidic conditions and able to essentially be within the tomato itself. It led to a tomato safety initiative that FDA put in place for Florida and Virginia-grown tomatoes. These were also contaminated in the late 2000s and had a significant impact on the California crop. Typically, the harvesting, the drying, the shelling practices can enable cross-contamination, so must be done very carefully. And since 2007, the USDA has required pasteurization of raw almonds to protect against the potential hazard. We also have seen a big salmonella outbreak attributed to pasteurized ice cream mix. In case, there were 80 cases which were reported to the Minnesota Department of Health, although it's estimated that likely thousands more cases occurred. It was associated with a certain brand of ice cream. The probable cause was identified as mix being transported in trucks that had previously hauled raw eggs. So one outcome of this was that the dairy industry and really anybody sending around tankers of food looks to verify tanker washing before accepting load that they're doing. But more recent time. Next slide, please. The outbreak of salmonella, actually there have been a couple in cucumbers. The most recent one affected at least 700 people and has 4 deaths. Here, illness is associated with cucumbers from a particular processor and one distributor. In this case, we weren't able to identify, nor the investigators were able to identify what the root cause was. But this is the second outbreak that has been linked to cucumbers with salmonella. The last one occurred just a year ago. So next slide, please. So some things to learn from outbreaks. Certainly, being diligent, being aware of sources that you might not suspect, because environmental sources of salmonella can be significant. We need to monitor plant environments, think about low moisture foods as an example, and perhaps cucumbers as well. We know that produce can be a source, and keeping aware of the regulations is key. Next, please. And this is another build slide. The first build should be to identify areas where strict salmonella control is needed. So it would be at the manufacturing level, but it could be applied to a food service area as well. So certainly, making sure that controls are in place around the cooking process and in any ready-to-eat areas. Being aware of water usage and not overusing water. Minimizing potential harborage sites, remove obsolete equipment, for example, and using the right sanitizers. This is bill number 4 on this slide. I hope I'm tracking effectively. For example, there are EPA-registered solid or powdered quat sanitizers that are used in manufacturing facilities, but there's liquid products as well. Have labeled claims against salmonella and can be used effectively against the strains. Following instructions for the use of sanitizers is important. And build on this slide is that there have to be effective controls, sanitation against salmonella. Slide, please, relates to salmonella transmission by ill food handlers. Because we know that this can and does occur. In fact, there was a study published a number of years ago that indicated a higher proportion of people, food workers, who were positive in outbreaks were positive environmental samples. At times, there were prolonged outbreaks in these establishments, and this was food service-based outbreaks, maybe hitting a percent reservoir of contamination. From this relates back to one of those contributing factors to illnesses is keeping ill food handlers, which be ill from salmonella, out of the kitchen. Next, please. Now, the specific food agent that I want to talk about is Cyclospora. This one is a little bit different in that it's a parasite, not bacteria. And a parasite, it does what it sounds like. It gets inside of a cell and essentially takes over that cell. And in this case, a Cyclospora cell affects human intestine cells. Also an interesting one as far as how it's transmitted. It is not transmitted from one person to another, but through the environment. Someone who is affected excretes these oocysts, or the little particles of cyclospora, in their stool, and then these basically hang out in the environment. And after days or weeks in the environment, they sporulate or they change shape, and this here of this red dyed substance shows one of these. Once they sporulate, they become infectious, and it's these infectious particles that can then contaminate food, like produce or water. When one consumes this food that might be contaminated, in 7 days, they will experience symptoms of watery diarrhea and other associated gastrointestinal related types of things. Next slide, please. From a historical perspective, we have seen outbreaks with cyclospora over the years. A notable one occurred back in the 90s, attributed to raspberries. The largest outbreak, recorded outbreak of cyclosporiasis, or illness due to cyclospora, at the time, within 1,400 people became ill in the U.S. and Canada after they consumed raspberries from Guatemala. It was hypothesized that raspberries became contaminated through spraying with various insecticides and fungicides that were used as part of the growing process that had been mixed with contaminated water that came from improperly constructed or maintained wells, which became contaminated with human fecal material. There's a fair amount of contamination because of the rainy season into these wells, and that was another hypothesis. And once the raspberries became contaminated, they remained such until they were eaten, because they really couldn't be washed thoroughly. If you think about the morphology of a raspberry, it's hard to wash it thoroughly. That this is a hypothesis because they never found cyclospora in raspberries. And then one where we have test methodology issues. The test is simply not sensitive enough. And the water and the berries were tested after the outbreak ended, so it could have been the right samples, just simply weren't tested. But moving to present day on the next slide, an outbreak of cyclospora attributed to cilantro earlier this year, with at least 500 cases reported. In case cilantro from a particular state in Mexico was applied to restaurants, at which people identified in illness. And there were some illnesses in these states also related to fresh cilantro. So import actions were put in place. So shipments of cilantro into the U.S. from Mexico are only allowed if proper documentation is submitted showing that the cilantro was harvested and packed in a state outside of the one that was implicated. It remains in place today. Next, please. Neospora is very, very difficult to control. It's resistant to essentially all of the common disinfectants. Really, no chemical will kill it. So control for cyclospora relies on knowledge of the source, why the import alert was put in place related to a particular area or source control. Also, water quality, such as that might be used for irrigation or was shown in the previous outbreak, fungicide spraying are very, very important as well. Next slide, please. The last example of outbreaks in the news, I talked generally about the category of improper holding temperatures. Because showed earlier, there's a number of contributing factors to foodborne illness. And improper holding related to that of proper hot and cold holding are crucial to the safe food, but often there are lapses here. What happens? Pathogens can persist and in some cases increase to harmful levels. Next slide, please. Some of concern for improper holding temperatures that I want to talk about include Clostridium perfringens and thrombotoxin. Slide, please. So Clostridium perfringens is found pretty commonly in raw meats, and it's really not an issue unless it grows to high levels. These cells are able to grow if there's poor temperature control in the meat products, like inadequate cooling. And this is caused by ingestion of large number of these cells when produce a toxin in the intestine, causing symptoms of and diarrhea. An outbreak occurred in 1993 when corned beef was produced and served around St. Patrick's Day. In this case, about 156 cases were reported, and found that all of these had eaten corned beef sandwiches from the same deli, and showed that the beef was cooked but then held at 120 degrees Fahrenheit when served throughout the day, and high levels of Clostridium perfringens cells were found in the beef. The illness is relatively mild, so we really see few outbreaks reported. My suspicion is because, frankly, very few people would go to medical care because their symptoms aren't too bad. But this is kind of a classic one of they would happen. Now, next slide, please. If we forward to present-day time and think about Clostridium perfringens. For this year, there's some salami that was recalled from a manufacturing facility, and at the establishment themselves did internal records review showing that the product had not reached safe temperature during the cooling process. It was not cooled appropriately. Now, no illness is reported as far as we know, but they could have occurred. And again, I think it's worthwhile to keep Clostridium perfringens and other temperature-related illnesses on the radar screen, because while these outbreaks can occur in food service, this example shows that manufacturing can also be a source, and attention to temperature control is key. Next slide, please. Now, a set of data we get from CDC relates to the kinds of foods that might be estimated to cause foodborne illness. This data, 2013, it was shown that fish products were the number one food to which illnesses were attributed. Next slide, please. Now, suspicion is that a lot of this is due to histamine production. And histamine is actually a chemical that's produced in fish after they're caught and die when they're not cooled appropriately. Fish are caught, they need to be refrigerated promptly, because warm storage temperatures can permit the native bacteria that are natively in fish to grow and produce an enzyme that reacts with also amino acids that are in the fish, this toxin. And you see here the kinds of fish that are most likely associated with scromboid poisoning because they have high levels of certain amino acids. So, mahi and tuna, for example. This is a very rapid reaction after consumption, includes intestinal symptoms, but also allergy allergy-like reaction. Now, it's not destroyed in cooking. It's undetectable to the consumer. And in 2001, there was a study done of outbreaks in ground tuna and found that outbreaks occurred in meat that had been thawed and but then was refrozen. And despite the fact that it was frozen and thawed a few times, the stromboli toxin was produced. And it's hypothesized that the grinding process could have raised the temperature a little bit, in the bacteria and causing the contamination to occur. So to combat issues around outbreaks that could be attributed to poor holding temperatures, there's best practices that need to be followed, but essentially it's following the proper cold holding practices for foods that might be held at room temperature. Cold holding practices include holding pan in cold wells that are filled above the top of the pan, or where issues can be if foods are not cooled to 41 degrees before placement in these cold wells, or if inadequate refrigeration space resulting in overstocking and poor air circulation. So there are certainly things that can be done in the places where food is served. But also there needs to be consideration, as was shown in Clostridium perfringens issue with salami, that supply chain needs to be considered as well. So we're getting to the end here, but next slide, I just wanted to highlight that Ecolab does provide information on a variety of microbial risks on the website, an update with links to information from public health authorities. So you might want to go to this site to find out more information about some of these agents we've talked about today, including Listeria, Salmonella or Cyclospora, or E. coli, or something that we were not able to address today. And I will now turn it over to Anna, who will direct questions to me.
Thank you, Ruth. Great presentation as usual. We've got several questions, and some of them are similar, so I'll combine them. The first question was, does pasteurization kill Listeria? And another question, which you probably would be able to answer together with the first one, how do you destroy Listeria in food? Can it be made safe?
Yeah. The good news is that Listeria is killed with heat. So typical pasteurization temperature, cooking of food temperatures that are recommended in the food code, for example, will Listeria. A raw food that might be contaminated with Listeria, like raw milk, through the pasteurization process will be made safe. The challenge is to guard against any recontamination of these foods that would be cooked to the Listeria, so there isn't cross-contamination of these foods after the cooking process that will contaminate it again.
Thank you, Ruth. Another Listeria question, and I am encouraging everybody to send more questions if you have them. But the next question, as I said again, Listeria related. The question is, can you talk more about genetics of Listeria and how the problem can span years? Is the problem spreading or not controlled, or not controlling and eliminated? So the question is it spreading or not controlled or eliminated?
Yep, and that's a very, very good question, and I don't have necessarily a definitive answer to it, because I think the reason I talked about whole genome sequencing is that we now have this technology that can help identify potentially strains that perhaps were in a facility for an extended amount of time, but more of that could have been that it existed in the past days and has come back. We need to learn more about the whole idea of persistence of Listeria. This is something in an area of active research. We need more data on the existence of these various genetic patterns of Listeria the environment, in facilities, in potentially food handling areas as well, to help us address these questions. And bottom line at this point, we have a lot of questions and we do answers at this point. But the fact that this technology is now available and being used will help address some of these questions over time. But as the questioner suggests, it does open up this whole area of questions as well that still need to work to address. I will say that I've attended one meeting at FDA at the last several months, and there's others coming up in the months to come that are attempting to at least ask the questions about this. So it's a very, very active area of research, and my best response to this is stay tuned and stay up on the literature, because this is one that is constantly developing these days.
Listeria is a popular topic. So one more question on listeria. What are listeria policies outside of the U.S.?
That's an interesting one. As I said, the policy within the U.S. is a zero tolerance, so no listeria can be detected in ready-to-eat product. If it is, it's considered adulterated. But outside the U.S., the policies are variable. Some countries, for example, permit levels of Listeria monocytogenes as high as 100 per gram in foods that do not support growth of the organism. This is based on the fact that it's generally assumed that high levels of Listeria cells are required to cause illness, and foods that don't support growth, such as something that might be frozen, or low water activity, or a low pH, those cells will not be able to increase to levels that could be harmful. So there is some variability around the world. This is one that we'll need to watch over time as the research is expanded with the advent of whole genome sequencing.
So we exhausted Listeria questions. There's another question. What is the second most common cause of foodborne illness? Since you mentioned the first and the third one.
Yep. Actually, the information that I presented was related to the cost of those pathogens. Norovirus is the number one most common cause of foodborne illness, and is the second most costly in terms of healthcare costs. Salmonella is number one, then norovirus, then Listeria is number three as far as a cost perspective. Listeria on the overall incidence of illnesses is actually pretty far down on the list, but because its outcomes are so serious and its mortality rate so high, it does have a very significant health care cost.
One more question related to Saccharospora. Can I wash dishes in 3-compartment sink with sanitizer or disinfectant, or wash hands with antimicrobial hand soap during the boil or situations?
Well, the challenge with Cyclospora, as I mentioned, is that there really are no chemicals that are effective against it. It's a very, very resistant organism. Parasites have a very, very resistant coat, essentially, around it that allows it to survive. So that you can rely on chemicals alone to inactivate Cyclospora. Certainly for a boil water alert, there's issues with the water, which could be a parasite contamination, and that water should not be used because it is not deemed safe. So if there's a way to use some safe water from an outside source, that's a possibility, but certainly not with the water that would be implicated in the boil water alert.
Thank you. I do not see any more questions coming, but I'm sure that as we finish our presentation, people would think, oh, I should have asked this or that. So we are encouraging you to send us more questions if you have any later, and we will forward them to Ruth, and I'm sure she would be able to help us with the answers. Meanwhile, I wanted to thank Ruth for a wonderful presentation and for your attention and for organization of this webinar by the team which helped us to arrange all of that. Thank you very much.
Ladies and gentlemen, that does conclude today's presentation. You may now disconnect.